I've had in my head my own made up version of the ABBA song. So long, serotonin. Nah, nah, nah. I've had ample time to make something up that maybe rhymes even. And yet, I just couldn't do it. I couldn't make it happen. This was a slog for sure. Fun slog. Great slog. But it was kind of like when I did dopamine, because dopamine has a lot of very existential sort of functions in the brain. Um, but at least with dopamine, they're very distinct and clear pathways, and each of them kind of have their own sort of, I don't know, main function, like the mesolimbic dopamine pathway. It's like, all right, well, that kind of goes through the limbic system and is heavily involved in reward cognition. Got that. Or the what is it? The neurostriatal pathway is heavily involved in movement. So serotonin, not so much. Serotonin, very different. Not many well-defined pathways, um, or rather, for sure many pathways, but not mapped out in the nice organized way that dopamine was. Ooh. I have many, much like the serotonergic circuits in our brain. All of my notes are very wishy-washy and going in many, many, many different directions. God bless serotonin, truly. Who is this fucking Sarah everyone keeps talking about? Who is this fucking serotonin? I want to beat her up. She's the new girl in town. Yes. God bless serotonin unless you have tummy aches all the time, in which case. Oh shit! It is happening. Thank you, Ender. You just, wow. Serotonin right to my brain, right there. Speak of the fucking devil. Oh my god. Well, let's see what the fuck. Who the fuck is this serotonin then? Who is this new girl? Um, we'll just get right into it. We can do it. You know, we'll just jump into the deep end. Serotonin is a signaling molecule, and it plays a role in a lot of different processes in the human body. So many, more than you can imagine, more than I even have pictured here. But of course, the brain and the GI tract are the main sites of serotonergic action, and you hear it in conjunction with dopamine a lot, uh, and it is similar to dopamine. Both serotonin and dopamine are monoamines. Can I make will my thing become a pen? Because they have this right here. They have a little amine group. Um, and serotonin is also like dopamine because it can act as both a neurotransmitter and a hormone for mega signaling. Um, so, and what that means basically is it facilitates communication within our central nervous system. It facilitates communication between our nervous system and our organs, and it can also transmit information through our bloodstream. It is a trait of all jacks here. Um, keep in mind throughout this entire fun time that we're about to have that the function of a single signaling molecule like serotonin depends entirely on its receptor, its location of action, and any other signals that are trying to cloud the information. Just file that away for now. %, Amy's here. 90% of our body's serotonin is found in the gut, specifically in the enterochromaffin cells that line our gastrointestinal tract. Serotonin actually used to be called enteromine. Enteric meaning intestines, of course. And this was because when it was first discovered by this guy here, by Vittorino Espamer, he first discovered it in the intestines. Probably not in humans. It was probably some other mammal that he found it in. And that was in the 1930s. That was a really freaking long time ago. Later on, there was another group of doctors, probably in the mid to late 40s, who were studying both hypertension, high blood pressure, and muscle contractions. And they found this substance that seemed to cause vasoconstriction, so a tightening of blood vessels. And because it was found in the serum and it affected vascular and muscular tone, they named it serotonin. So that's where the name serotonin comes from. Found in the blood and affects vascular tone. And right here, this is Dr. Report. He was on the team who was studying hypertension specifically. And they first isolated the neurotransmitter, or in this case, the hormone, from animal blood. And it's kind of interesting because this was back in the day. There wasn't like an easy way to kind of do this. And this was in Cleveland or something. So they went to this Cleveland slaughterhouse and they would get gallons and gallons and gallons and gallons of blood and pour it into these garbage bag lined barrels that had what's it called, cheesecloth, to sort of filter it. And that's how they first isolated serotonin. He was a nerd. Look at that guy. He's not even looking at the camera. He is way too focused on what he's doing. He's a dork. Later still, there was, because the remaining 10% of our body's serotonin is in the CNS. So at the time, this woman, Dr. Betty Twarog, was studying mollusk muscles. And she had figured out the substance that caused their muscles to contract. I think it was probably acetylcholine. But she was having trouble figuring out what the substance was that was making their muscles relax. And she knew it was a neurotransmitter, but at this time, this is like like I said, like late 1940s, early 1950s, neurotransmitters were not, they were theoretical. We weren't really sure that they existed at the time. But even still, she took her unknown substance that she had isolated and she compared it with samples of serotonin from the previous guy's lab. She determined that they were the exact same. So she not only proved, or rather identified serotonin as a neurotransmitter, but she also proved that serotonin and this enteramine were the exact same substance. Why do we call it serotonin? Well, the first drug company to synthesize serotonin decided that that is what we would call it. I mean, enteramine makes a little bit more sense considering where the bulk of it is, but serotonin sounds again like the new girl in town, so it's a lot funnier. All right, so as I said before, serotonin is a monoamine. Who is Sarah? God, we still don't know. We still don't know, do we? Who is this Serotonin. I actually, because my name, of course, in a lot of places is dopamine. I do see a lot of serotonins, like people who spell it Sarah tonin, and I'm like, hey, we're cut from the same tree, monoamines and all. Um, okay, so serotonin is like dopamine, which means it's also like epinephrine and norepinephrine. Um, so they all get cleared from our system by the exact same enzyme, MAO, or monoamine oxidase. Serotonin, though, is made from a different amino acid than dopamine and all those other guys. They're made from tyrosine. Boring. Serotonin comes from tryptophan. Way cooler. um, so the tryptophan will get converted into 5-HTP and then later into serotonin. This is the biosynthetic pathway. Serotonin in the CNS is completely separate from serotonin in the periphery because serotonin can't cross the blood-brain barrier. It can't. There's nothing that can carry it into the central nervous system like that. However, tryptophan and 5-HTP can cross the triple B just fine. So the brain still has a way of making its own population of serotonin. So obviously, serotonin in the gut versus the brain has very, very, very different functions. Although, as we'll see in a bit, the gut is in constant communication with the central nervous system. So the location of serotonin's actions are important, and it'll do a lot of different things depending on whether it's acting on smooth muscle, for example, or on peripheral nerves, or some region in the brain. The outcome of its action is going to be very different depending on those things. We're remembering. We're keeping that locked away in our brain. This diversity of function has almost entirely to do with serotonin's receptors. There's a lot. There's like 14 of them. And their type and their density and their location determine serotonin's function. Location, location, location. Very, very important. But of course, in both the brain and the digestive system, serotonin is a signal. That is its job. That is who it is. This is just a little summary of the functions that we may come across in these two areas. A lot of its CNS functions have to do with regulating other neurotransmitters like dopamine and acetylcholine, and also the two very important the excitatory neurotransmitter glutamate and the inhibitory neurotransmitter GABA. Serotonin affects the release and the expression of those two neurotransmitters. It's very important. We'll kind of dive into that later. But also serotonin is important for the more esoteric functions of the brain, like learning and memory and cognition. And the destinations of the different serotonergic innervations in the brain can also affect our mood and our emotions. That's for later, though. The brain is later. Because we're diving in gut first. Just as God intended. And maybe I will even show you why vomiting is considered a central and a peripheral function. Maybe. We'll see. We'll have to see. Okay. Wow, this feels swimmingly so far. It feels swimmingly. That's a saying. It feels swimmingly. It feels good so far. It's very normal. uh, it's just serotonin, or dare I say, enteramine in the gut. It makes so much sense. It's so it's so lovely. I'm just gonna bask in this. um, Serotonin's action in the gut, mainly the small intestine, perfectly demonstrates its job as both a hormone and a neurotransmitter. Your gut, as you may know, actually has its own devoted nervous system called the enteric nervous system. Uh, and it lies between different layers of our intestines. So if this right here is different layers of the small intestine tissue. Um, we have the submucosal plexus. A plexus is just like a networked. It's a nerve network basically. That's very interconnected. So the submucosal plexus is the part of the ENS in the small intestine that regulates secretive functions. Whereas if we go a little bit lower to the myenteric plexus, this more has to do with reflexive muscle contraction, so peristalsis in the small intestine. The enteric nervous system is autonomous, but proper and healthy digestion does require communication with the central nervous system. And it does require innervation from other branches of the autonomic nervous system. This is my amazing spinal cord. Amazing. Our gut gets sympathetic input. So that's fight or flight. These nerves are on all the time, but they're known as the fight or flight kind of nerves. Our gut gets sympathetic input from these nerves, and it also gets two parasympathetic inputs, or rest and digest, from the pelvic nerve and the the vagus nerve, the very famous vagus nerve. So these are all, they're not a part of the enteric nervous system, but they sort of shake hands with what we've got going on, specifically in that submucosal plexus and the myenteric plexus. But don't worry too much about what these branches do, because we just care about digestion. Oh yeah, this is just the breakdown. The ENS is technically considered a branch of the autonomic nervous system. That kind of makes sense to me. Yeah. Yeah. We'll remember that. All right, so here we go. When you become just absolutely impregnated by a super burrito, your stomach digests it for a little bit before it moves into your small intestine. Here in the small intestine, enterochromaffin cells, or EC cells, have these special receptors that are sensitive to chemical and mechanical changes that are brought on by eating. So, of course, the food that you eat is going to change the chemical environment, and it's also going to cause pressure and distension on your intestine, both of which can be detected by these enterochromaffin cells. They also happen to have, oh my god, all of the components for making serotonin. They have that good old tryptophan hydroxylase to turn tryptophan into 5-HTP. They have that good old aromatic amino acid decarboxylase, I think I said that right, to convert the 5-HTP into serotonin. And they have VMAT, which releases serotonin from its stored vesicles. All the things that we need are right here. What's a burrito? Oh man. A burrito is a small donkey. Is a small donkey. You're not wrong. Amy's not wrong.
I'm always talking about getting impregnated by a small donkey.
Yeah. Oh my god. Disgusting. Oh my god. So, when these EC cells sense the changes brought on by this burrito impregnation, is impregnation a word? Um, they release serotonin into both the lumen, or the inside of the tube, the inside of the intestine, as well as into the tissue layers below the cell. So here, serotonin inside the tube is going to act as a paracrine signal, so a very short range signal. It's going to let the cells around it know that it is time to release water and electrolytes and mucus into the lumen of the intestines in order to help, this is gross, but like slide the things being digested along. Disgusting, but cool. The serotonin that's released into the tissue will bind its 5-HT3 specifically receptor on certain neurons. These neurons right here are called intrinsic primary afferent neurons. So they're just part of the ENS, and they're going to be part of a reflex arc. So they synapse onto some interneurons, and then these interneurons will synapse onto motor neurons. And the motor neurons will stimulate muscle contraction. This one up here, this is the, what's it called? It's called, I think it's just called the circular muscle. So that those motor neurons will stimulate this kind of squeezing muscle contraction, whereas this down here is the longitudinal muscle. So that will stimulate muscle contractions that kind of go in waves along the tube. Yeah, it's not good to eat small donkeys. They're probably not good for you, but they look so tasty. Remember that the GI tract also gets parasympathetic innervation from that massive vagus nerve. The vagus nerve is it's very well known because it's so, not only is it huge, but it's a mixed nerve. It can be sensory, like the one that I'm about to show you here. It can sense things, and then it also has motor branches, so it can bring signals to muscles to cause movement. But here, serotonin is also going to bind to that same receptor, 5-HT3, on vagal afferents. Afferents take things to a place. They go to the CNS. Means to. If you speak Spanish, that's literally how they teach it to us in school. It's the opposite of efferents, which exit the central nervous system. So this is taking information to our brain. They take the information to the brainstem to kind of communicate things like the distendedness of the intestines. It brings information about pain, and it brings information about potentially poisonous stimuli to the brainstem. Okay, the vagus nerve just so happens to synapse with a little place called the vomiting center, which integrates signals from those poisonous or noxious stimuli. Also from like overindulgence. If you overeat, that can also potentially stimulate this pathway. So serotonergic information of the vagus nerve, which innervates a lot more than just the small intestine, let's not forget, can lead to reverse peristalsis and expulsion of digestive contents. Again, as a probably evolutionary way of ridding really quickly and irreversibly almost, ridding the body of poison. That's, yeah. This was just a really good depiction of the area postrema, that little dot. But then it's kind of a weird, because I'm like, all right, I see the striatum and the hippocampus, the amygdala, And then in the middle, I'm like, what are they trying to tell me?
Yeah, what's the big blue part?
It could be like the thalamus. Like, yeah, because like oh my god, you are freaky correct. This just really looks like Lake Superior.
It's just like the Great Lakes region. Everybody has a little Great Lakes in their brain.
Yeah, or it looks like a crow that's like,
Are you wearing neuron earrings?
yes. Oh my god, I forgot. I have neuron earrings, courtesy of my
Cute.
sister. Um, yeah, wow, really, I, I, I'm like, this is where the pituitary might be. The amygdala actually could be up here. I have no idea what they could possibly, what this is supposed to be. It's very vexing. Um, but it was a real, the area postrema, if you search that, it's a lot of actual brain stems of like human cadavers, which is cool, but kind of gross and kind of hard for me to point out where it is. So this was, despite the blobby blueness that I don't understand, this is a really good representation of the area postrema. Um, another part of this, so the vomiting center, it is indeed a thing inside your head. You have a big, just like a big blue lake inside your head. um, the vomiting center is like located within a specific nucleus of the brainstem called the solitary tract. And another sort of part of that system is the chemoreceptor trigger zone. I like how that sounds. Um, and that part of the brain is very interesting because it's outside the blood it's part of the brain, but it's outside the blood brain barrier, which is important to know for this next little interesting tidbit, because in addition to stimulating the vomiting center through the vagus nerve, serotonin can also travel there all by itself. And it can stimulate that same receptor 5-HT3 on the cells in the area postrema. So if there's something that's really, really poisonous, like if you ate something that's just really not good for you, EC cells will produce like a fuck ton of serotonin, like so much, and it'll accumulate in the bloodstream. And normally, serotonin in the bloodstream will get snatched up by platelets. And that's a little fun fact. Platelets have serotonin in them, and they bring it to places that require clotting or wound healing, and I think serotonin is supposed to aid in that process. But in any case, um, the if there's more serotonin accumulating in your bloodstream than the platelets can go around and snatch up, then it will build up and directly trigger the vomiting center. Hence, this process of normal digestion or responses to overindulging and pathogens in the GI tract involve both peripheral and central nervous system structures. It's fun. So serotonin, we may call it the happy molecule. It might be considered a good mood molecule, but not for anyone with IBS or other inflammatory bowel diseases, because it's actually possible that these individuals have overreactive EC cells that produce excess serotonin for seemingly no reason, and therefore they have increased amounts of serotonin in their gut. So to them, serotonin is more the diarrhea molecule than the happy molecule. Can I have some more water, please? All right, real quick, we're just gonna take a look at this summary of the serotonin receptors. All tasty. There are 4 families of serotonin receptors. We have 1, we have 2, 3, and other. They all have their own personalities, and some of them even, counteract each other. What's not reflected here is the whole kind of location thing, in that some of the effects may be different depending on if the receptor is in a different part of the brain. Um, so, for example, like anytime you see neuromodulation, that kind of indicates that this receptor can lead to either an increase or a decrease in some other neurotransmitter, depending on the brain region involved. Um, but uh, that's, we'll only get into that a little bit. And then over here, these are important because this is sort of what separates the different families from each other. So 5-HT1A, for example, is very interesting because it is coupled to GI, which basically that just means that the activation of this receptor leads to a decrease in cyclic AMP, which is a second messenger. So the overall effect is inhibitory because its activation decreases the creation of something that would otherwise propagate a signal. And yeah, both of the HT1 receptors are like this. The HT2 receptors are a little different. They there's their signaling process is a little more complicated than just like inhibiting or stimulating something, but their ultimate goal is to increase the amount of calcium, which is also a second messenger. So not only will the calcium go on to sort of do its own thing and maybe activate or inhibit some other proteins, but because it's a positive ion, increases in calcium tend to lead to more neurotransmitter release. So we can say that for the most part, these are excitatory rather than inhibitory actions. But I just learned what the scroll wheel does. But yeah, the different signaling mechanisms of these receptors are very, very important, but I'm not gonna 100% like go into them in detail because I'm not here to stimulate your chemoreceptor trigger zone. We'll just, we'll come across it organically. I do really like these earrings. I should wear them more often. Okay, I lied a little bit. I want to talk about the 5-HT3 receptor, which you've heard me say a lot already because it is in your ENS nerves, it's on the vagus nerve, it's in the frickin' vomiting center, and it's weird. I don't know if you can tell by this chart, but it's 100% different from all of the other receptors here. It's not coupled to a G protein. It's a frickin' ion channel. It's a cation channel. So when serotonin binds this receptor on a neuron or whatever, it's a channel. So it opens and it allows positive ions like sodium and potassium to enter the cell, to enter the neuron. And what happens when we make the inside of a cell more positive? Say it with me. We get a motherfucking depolarization. Okay, depolarization. I can go over this really, really quick. It's not, it's complicated, but not really. When the membrane depolarizes, that means that the signal, the electrical impulse, is going to be propagated along the neuron to wherever it needs to go. And it also means that neurotransmitter is going to be released. That's just what happens when you have a lot of positive charge inside of a nerve terminal, kind of like I was saying with that GQ mechanism and the increase in calcium. calcium is a very positive cation, so increased in intracellular calcium tends to force neurotransmitters to be released. Um, so this receptor is excitatory, 100%. When something stimulates this receptor in the vomiting center, maybe the vagus nerve is upset by the presence of a pathogen or a chemotherapy drug. Drugs directly bind this receptor in the vomiting center. Um, or and what's that third thing? You're wasted. Alcohol. Alcohol actually can also directly bind this receptor in the brainstem, and alcohol is a stomach irritant, so it could also, both. um, but when those things happen, you are gonna throw up. These excitatory signals caused by serotonin binding to this cation channel can't be stopped. They bind, and those vagal efferents are gonna reverse peristalse the shit out of you. It's an all or nothing kind of deal. you, you knew it. The alcohol thing, if that's what you're referring to. Yeah, it's like, wow, that makes sense. um, yeah, you can't stop it unless you block it. Plot twist. You can block it. You can, actually. 5-HT3 antagonists being things that bind receptors that totally block its action, are some of the most effective antiemetics. Things that make you vomit faux show, like chemotherapy and opioids, are totally counteracted with 5-HT3 antagonists like Zofran here. It's also used to treat morning sickness. I mean, it completely blocks that depolarization from happening so that the noxious stimuli signals don't get transmitted to the muscles involved in vomiting. Boom. Um, in in addition to the vagus nerve and the vomiting center and some other brainstem areas, 5-HT3 can also be found in other parts of the cerebral cortex, like some limbic areas, which are involved in emotional cognition. And it actually helps release dopamine in the ventral tegmental area, um, which is a part of the reward circuit. So this receptor's presence in these structures, along with some other subtypes, shows that they also mediate not just serotonin's physical kind of evolutionary effect on like vomiting, but also on serotonin's more esoteric and brainy functions. So we're done. No, we're done with the 90% of serotonin in the body. Now we're gonna hang with the 10%, the complicated group. So CNS serotonin comes from structures in the brainstem called the Rafe nuclei. There's a couple different ones, and they go to different areas, but generally, neurons go up into brain, and they go down into spinal cord from this serotonin origin in the Rafe nuclei. Usually, a neurotransmitter will exhibit its effects by being released by a presynaptic nerve terminal and then binding to a postsynaptic nerve terminal to transmit a signal. And then that section in between is the synapse. That signal that's being transmitted will either increase or decrease subsequent neurotransmitter signals depending on the receptor. For example, fun little guys called 5-HT1A receptors are on the presynaptic membranes in the Rafe nuclei, um, and on postsynaptic membranes elsewhere in like the prefrontal cortex. But the ones on the Rafe nuclei, where serotonin is originally being released, we call those autoreceptors because they're responding to serotonin that's being released by their own cell, basically. Um, and when serotonin binds them on that binds those autoreceptors, this leads to a decrease in subsequent serotonin release. This is negative feedback. It regulates itself, basically. We love that. We love negative feedback. This is partly why SSRIs like Prozac here don't help right away. You don't get prescribed antidepressants and then you take them and you're better, unfortunately. Because what happens is they do increase serotonin. You know, that is an immediate effect. They do increase serotonin by preventing them from being cleared from the synapse by the serotonin transporter. So that's why we call them selective serotonin reuptake inhibitors. It keeps serotonin in the synapse where it's going to have its nice functions. However, when that happens, those autoreceptors are going to keep doing their job. They're gonna keep regulating themselves like they're supposed to do. So that initial like increase in synaptic serotonin will lead to just like a really quick clearance. And the receptors will need a few weeks in order to desensitize before an actual rise in serotonin in the synapse can dole out its therapeutic effects. So let me repeat some of that because I know it seems a little weird, but uh, your body has like set points basically. So you can think of the autoreceptors as having a sort of set point for the amount, like the level of serotonin that it reaches before it's like, all right, you're too much, get out of here. So if you're really depressed and your depression is based on really low serotonin, then any little tiny increase in serotonin toward what's actually normal, your body is going to see that as, oh no, I need to do negative feedback. I need to get this out. But luckily, after a few weeks, those receptors can be desensitized. They can be sort of taught a new set point, and they will stop being so quick to do this negative feedback mechanism on the rising serotonin levels. Um, so, and Prozac doesn't even like Prozac doesn't bind this receptor. Prozac interacts with SERT, the serotonin transporter. It doesn't even interact with this receptor. So even without these two things interacting, like I don't know, medications that act just on the system can bring about these uh, changes that are very indirect but also very profound. There are very complex relationships between signal, receptor, brain region, and models of anxiety and depression can kind of shed some light on those things. Um, I don't even need to switch receptors yet because 5-HT1A is just everywhere, and it generally I'm going to preface this by saying that it generally has an inhibitory effect on its effector areas, on the places that it goes to do things. As you kind of saw with the autoreceptor. But that doesn't always mean that the end result is inhibition. Don't worry, is the first thing I have to say. What you're looking at is a representation of how different amounts of serotonin affect the existence of anxiety phenotypes. So, let me just show. On the left, it's telling you where we are up here. We're in the Rafe nuclei. What's the Rafe nuclei? That's where serotonin originates. So this is the This is a neuron that will release serotonin originating here. And we're going to the prefrontal cortex, which is a part of your brain that is very involved in all forms of like cognition and learning and emotion and things like that. Um, so this is a serotonin neuron, and it synapses. It connects with both. Both a GABA neuron right here. GABA is an inhibitory neurotransmitter. When it binds its receptors on a neuron, it decreases the activity of that neuron. It dampens signals. Glutamate, the green, is the opposite. Glutamate is excitatory. It facilitates communication between neurons. And then lastly, just notice that this glutamatergic neuron goes back to communicate with the serotonin neurons in the Rafe nuclei. So in this model, we have 5-HT1A receptors. They're 5-HT1A receptors are on both of these guys. They're on the GABA and the glutamate. And we know that in general, 5-HT1A is inhibitory. But, oh man, so many. Brings me back to the basal ganglia. In this model, the 5-HT receptors, the end result is they're excitatory because they bind, serotonin binds 5-HT1A on the GABA neuron, on the inhibitory neuron, and it inhibits it. Serotonin inhibits the inhibitory neurotransmitter. So, and serotonin preferentially binds this guy way more than it binds the glutamatergic, where it does have an inhibitory effect. It's being normal there. But because it preferentially binds this, we call this disinhibition. The blockade on the glutamate receptor caused by the GABA receptor is lifted when serotonin binds 5-HT1A. I know that sounds really weird. The concept of disinhibition is kind of mind-fucking. I know I had this problem talking about it in regards to dopamine and the basal ganglia and how it, there's disinhibition pathways in the basal ganglia, and then there's disinhibition pathways too. It stimulates my chemoreceptor trigger zone. No. It's a roller coaster, and this is brain circuits, but it's fine. It makes sense. So let's just go through the differences in serotonin and how they may or may not cause anxiety. So in the very top diagram, we have no serotonin. There's nothing. So the GABA neuron is free to inhibit the shit out of the glutamate neuron. And glutamate being, it's going to other areas in the PFC. That's what this is trying to say. Is it's going to, it's going off into the distance to mediate its cognitive effects or whatever. When we're inhibiting that glutamate neuron, we are decreasing the amount of communication that is going out into our prefrontal cortex. And the result is a no anxiety model. This is consistent with our understanding that anxiety tends to be associated with too much activity in the prefrontal cortex. There's too much going on there. Anyone who has anxiety can probably, see the logic in that. Overthinking. There's too much going on. So that's why no serotonin. That's great for anxiety in this specific model, in that specific area. With low serotonin, we do see some anxiety because now that we have serotonin to bind the inhibitory neuron, um, the inhibition is lifted a little bit. And because, remember, the location, for whatever reason, serotonin prefers to bind 5-HT1A on the GABA neuron versus the glutamate. It just does. I don't know why. But the effect that it has on the disinhibition then is going to be more profound than its effects on, directly inhibiting it. As a result, you're going to have more activity in the PFC versus the no serotonin, and you are going to get an anxiety model. Normal serotonin, well, we have the perfect balance. We have a wonderful amount of serotonin lifting the inhibition on the glutamate. We have a good amount of serotonin inhibiting the glutamate. So, butthole kiss. The perfect amount of this excitatory neurotransmitter is going into the PFC, and no anxiety. Lastly, of course, we have high serotonin. Serotonin levels that have increased too much will cause anxiety because, again, it's this freaking preferential binding here. It's really lifting this inhibition to the point where, and you do see it a little bit in the normal serotonin, but it's to the point where the neuron is still being uninhibited enough that it will go back and release glutamate, an excitatory, again, neurotransmitter, on the serotonin. So this is positive feedback. The more serotonin is being released, the less inhibition there is on the glutamate, to the point where the glutamate backtracks and continues to increase the serotonin. Hence, we get too much activity in the PFC, anxiety. This is massive right here. This is huge. And I think I might go into it a little bit later, but different like SSRIs and different drugs for psychiatric conditions, you'll notice that certain ones have like, they kind of will bind this receptor, but then they'll block this receptor. And that's good because those receptors have sort of opposing effects anyway. It's really, I was very much wowed by this model. It was really, really, really cool. And what's anxiety without depression? So this, again, so freaking cool because with no serotonin at all, we were chilling on the anxiety. But obviously, if you don't have any serotonin in the system, you're going to get depressive symptoms. I guess the easiest way for me to explain, let's go through it first, really quick. So we're experts now because we looked at the anxiety models. So depression is obviously observed when there's no serotonin, and therefore the glutamate is overly inhibited by that GABA neuron. And while anxiety may not be associated with underactivity, depression clearly is, because those areas are involved in emotional regulation, and they also communicate with the reward system. Oh my god, so many things to consider. The PFC isn't just like an end result, like it's communicating with other areas of the brain, and a very central symptom of depression is anhedonia, or the inability to feel interested in or motivated by anything. And deficits in the PFC and its communication with the reward system are implicated in it. So obviously, it makes perfect sense here that no serotonin is going to lead to depressive symptoms. Low serotonin is the same way, except here, this is where it's similar. So this leads to models of both anxiety and depression, comorbidity. So with anxiety, we saw that uneven regulation of the glutamate neuron that led to overactivity. But with depression, the threshold, I believe the threshold for those functional connections to the prefrontal cortex is still too low. And that is evidenced by the fact that depression is not observed at all in normal and high levels of serotonin. Um, so cool. So when it comes to these higher level, these cortical connections in our brain, it can be hard to parse out the neuron types, the neurotransmitters, the receptors, the regions. And there's more going on here, obviously. It's not just this one GABA neuron and this one glutamate neuron. But the main takeaway is that differing levels of serotonin in a system lead to differential receptor activation and preferential neuronal activation. Oh, don't worry, I'll talk about drugs and then it'll be even more clear, I swear. Uh, so, and this, like I said, this is just the Rafe nuclei and the PFC. You saw in the other picture, the Rafe nuclei, oh my god, it has projections all over the frickin' brain. So, saying that serotonin plays a role in these cognitive functions isn't incorrect, but even the subdivisions of cognition are arcane. God. Very arcane. Like memory. Memory! Is that from Cats? I only know memory. I don't know any other part of it. Amy's got more than me. She does?
Like, yeah.
I've never seen School of
Rock.
Rock.
She sings that much.
I know.
What?
I know, I've never seen it.
She sings that much and Jack Black goes, okay, stop.
Now I really need to watch it now, I feel. It's in Jersey Girl. That's what makes me think of it. Anders. I have summoned Anders with cats. That's what I've heard. I've heard that there are still badonkers in it. But all I know about it is it's a really good movie. It's very cheesy. But it's a Kevin Smith movie called Jersey Girl. No! And I made you watch it. And the girl has to do she has to pick a song from a musical for like a talent show to do with her dad. And when it comes time, the lady announcer's like, and now, blah, 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 doing Memory from Cats. And now, blah, 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 doing Memory from Cats. Like, everyone does that song. And then the main character, the little girl, does the Mr. Something's Meat Pies, the Sweeney Todd.
From Sweeney Todd.
Yeah, it's great. Memory! Okay. It's a cognitive function. It's not confusing. There aren't a million different types of memory or anything like that. And it's not super intertwined with a ton of other processes. It's so easy.
Remix the Meat Pie song as Mrs. Lovett's feet pics.
No! Mrs. Lovett's feet pics. Oh man, I wish I could erase that from my memory. Okay, memory. You need a memory in order to learn. And as you learn, you adopt certain behaviors, which can involve movement and language or thought. And in general, these processes are tied to emotion. And the structures that help you do these things make up the limbic system. This is more or less the limbic system right here.
Thank you, Anders.
Oh my god. He's put it together for us. Oh my god. Mr. Love is sweet fix. Come and get your treat fix. True, sad, true. I don't remember the song.
Mrs. Love is sweet fix. Come and get your treat fix. True, sad, true.
I'm standing up.
Look at Mrs. Lovett's feet pics.
Doesn't Helena Bonham Carter play?
Yes.
I could look at her feet. Just saying. Anyway, um, God, uh, limbic system, emotion, memory. These processes seem like they're so high order and special, but they're not. Because look at them; they're in the inside of the brain. They're carnal. They're not up here. This is where the big guys go. They're not cortical. They're they're very deeply ingrained, and I don't want to say automatic, but like innate, kind of natural. Uh, so it it makes these snappy learning things happen, and then the cortical structures integrate them and add like context and shit to it. Um, but the two structures that we care the most about in the limbic system are the hippocampus and the amygdala, which uh, I don't want to go back, but the other picture had oh my god. What am I trying to say? What's a short version of a word?
A short version of a word?
Yeah. An acronym? No.
No.
Shorthand. I'm just gonna say
An
shorthand.
abbreviation.
Abbreviation! Abbreviation. What the? Thank you! Jabba the Butt? Oh my god, you came at the perfect time. Abbreviation. Fuck. My lexicon recall. Amygdala was abbreviated as just Amy. It was really funny.
What my name is short for.
Amygdala. Oh my god, please.
Like the girl in Star Wars, Princess Amygdala.
All right, Princess Amygdala.
Mm-hmm.
Well, your Princess Amygdala is a part of the limbic system, and it encodes, as in like kind of creates, but also stores memories, and consolidates them. Specifically, like ones that have a lot of like emotional arousal, whereas your hippocampus basically just encodes. It doesn't really store memories for very long. It sends them other places. But the hippocampus is very important for committing things to memory in a more like instantaneous way. And these two structures, of course, communicate with each other. If an event is associated with a strong emotional context, the memories of that event are going to be way easier to retrieve thanks to this interplay. Like, if you, I don't know, let's say if you remember like everything about a specific day, even like the date, like on April 1st, whatever, all this stuff happened, blah blah blah. People are like, how do you know so much? And you're just like, I have a good memory, but in your head you're like, I shit my pants on the bus that day, and it was very traumatic and emotional, and now I remember every single thing about that day. Amygdala! That's basically what's going on. And I mentioned fear. The amygdala is involved in fear extinction, which is where you learn not to be afraid of something that you don't necessarily need to be afraid of. Them's the areas. Let's look at the receptors. Oh, we already had them. They were already up there the whole time. Whoop. The entire time. 5-HT1A. I know we're sick of her, but she's important. It is in both of these limbic structures, plus the neocortex. Neocortex is the seat of cognition. It is our galaxy brain. It is very evolved. And the presence of 5-HT1A in the neocortex means that it probably mediates cognition. I mean, probably. And as you just saw, cognition can originate in the lower structures of the limbic system. In the hippocampus specifically, 5-HT1A actually impairs memory by inhibiting glutamate and acetylcholine neurotransmission. We remember that 5-HT1A is inhibitory. We remember that. Both of those neurotransmitters, glutamate and acetylcholine, or ACH, are needed for memory formation. Many treatments for dementia, actually, are medications that prevent ACH breakdown. as a way to preserve memory function. Um, and this am I a professor? No, not really.
She is on the
I am on the internet.
internet.
I have a degree in molecular cell and developmental biology, and I like to teach people and talk about those those things. And people actually voted on serotonin. I almost didn't want to do it because serotonin is it's a spicy meatball. But yeah, I just like to research and I like to talk and I like to teach and I like to converse. Um, are you are you interested in biology at all, or are you a professor? Are you a professor? Or in school maybe? Um, okay. Will you hand me your water before you go? Um, 0 out of 10 recommend. No, I'm sorry. Uh, did you have like an interest in it when you started though? Like, do was there initially some passion there? That means when I say agonist and antagonist, you're like, ugh, God. That's really cool though. Even though you don't recommend, I think it's pretty cool. I love the subject matter, and I love reading about it and learning about it myself. And a lot of times I feel like even though I'm here to like share the information and maybe teach people, it helps me obviously. Like it helps me like conceptualize and learn things. Uh like my particularly I actually did a lecture on acetylcholine and sleep, and like mid lecture I like had a brain blast. I was like, "Oh my god, wait. This just made me realize how this works. It's great. It's great." Okay. Oh. Now everything I said, I feel, I swear serotonin doesn't give you amnesia. I promise. Like it doesn't. That's not like the main thing that it does. Uh but that's the thing about regulation. Some things do this and some things do this. Me 2024, some things do this and some things do this. Um but yeah, that's the nature of regulation. There's a ton of different receptors and areas that are involved in this process. Some do the opposite, like 5-HT2A, as you may have seen here. Thank you, thank you. I it would be fun as like a, it's not like doctor, where it's like you cannot call yourself a doctor. But I don't know how mad professor people would get if I was just like, yes, I heard Professor Dopamine at your cervix. Like I don't know how appropriate or inappropriate that is, but that would be fun. I feel like my Halo name somewhere, like my Microsoft Xbox thing is Professor Dopamine. No, it is actually Dr. Dopamine. But that's actually a joke. Dr. WD-40, that sounds awesome. I'm actually gonna make a note of that. Dr. WD-40. Do they do lectures? I always have to say at your cervix. It's kind of a curse. It's inappropriate. Um 5-HT2A, opposite of 1A, in here at least, because it is excitatory. It promotes communication within these areas, and it signals with calcium. And remember again, calcium is a positive ion when we have lots of positiveness inside of a cell, it causes the release of neurotransmitter and or the propagation of a nerve impulse, of a signal. Oh, that's cool. That's like that's like a nice, well-rounded bunch of things there. I will absolutely check them out. My brain deleted the W and I thought it said DRD40 and I was like, another dopamine receptor. It's bad. Oh my god. So because of this excitedness of this receptor, glutamate release is increased in the hippocampus, which helps um, in the creation of memories, specifically object memory, which has to do with our natural propensity to explore new things in our environment. Um, and it is super expressed in the cortex, the cool hot dog, not like hot dog, but like hot dog parts of the brain, as well as the visual cortex. Not your eyes, but like your visual cortex. um, oh yes, fear extinction is actually what is mediated by 5-HT2A. So what I mean by that is when serotonin activates 5-HT2A in the amygdala, this facilitates this process where you become less and less fearful of a stimulus, usually of a stimulus that you don't need to be afraid of. So the opposite would be people who like don't have an amygdala have an inappropriate amount of fear extinction in that they have fear extinction for things that they should be afraid of, kind of thing. But in this case of the normal fear extinction, this could make 5-HT2A a therapeutic target for certain anxiety disorders like phobias, where very non-fearful stimuli cause a fear response. So, this is a very interesting takeaway of these receptors in the limbic system, that they counteract each other, and it again, it might seem like memory impairment is a bad thing, but it's a regulation thing. It goes up and down. It's not gonna only cause a complete and total amnesia. um, 5-HT2A is actually like a really hot ticket receptor for yeah, Jabba the Butt. Thank you. I hope the receptors talk is not too pharmacologically triggering. um, if you don't mind, I'd be interested to know what you work on. What was I saying? Hot ticket? Yes, 5-HT2A. Again, very widely expressed in our prefrontal cortex, parietal cortex, somatosensory cortex, all of which are involved in very high order brain processes like sensory perception and spatial reasoning. It is also found in the orbitofrontal cortex, which is involved in decision making. And then, oh my god, the visual cortex once again, which is needed for seeing. Sight perception.
Foreseeing, like, see
the future? Yes. 5-HT2A activation in the orbitofrontal cortex makes you see the future. Oh, retail. Oh, so you are you like counting pills and like talking to people about their medication and being like
careful with this Tylenol.
Yeah, careful with this Tylenol. You gotta take birth control every day. Oh, that's all right though. That's all right. Um, OFC, visual cortex. You need to see. Did you know this? You need to see usually, usually in order to make decisions. It helps at least. It helps. And remember, lots of variables at play when it comes to a neurotransmitter's effect. Uh, Receptor, cell type, location, other neurotransmitters, extrinsic signals, outside of you signals. Oh god. Yeah. I don't I'm in a region that is very tech bro, and they they're kind of flooding the market when it comes to like certain stimulants, and it's been a problem. There was like a huge shortage. There's been a problem for people that therapeutically uh need those stimulants, and it's like, yeah, I feel that feeding addictions, just giving people things that are, yeah, literally, yeah, everyone's on Adderall for one reason or another. They're like, I want to not eat. I want to pay attention. Like, does it really make you pay attention though? Are you paying attention? Or are you just like, did you pay attention and do your homework, or did you just organize your magic cards for 8 hours? Is that paying attention? Yeah, that's a common one. It's methamphetamine? Hold up. No way. Hold on. I am like so curious. Oh my god. Cuz people call Adderall meth, and I'm like, no it's not. Shut the fuck up. Like, I get it, but we don't need to stigmatize further. But yeah, holy shit. They literally give meth. They literally prescribe meth. I had no idea about this. Oh my god. I wanted to show me the chemical.
Brand name.
Yeah. Yeah, well, I
Who
want to
makes it?
see.
Who makes the brand? Who makes that brand name?
Ooh, let me see. Oh, it's not telling me the manufacturers. Oh my god. Ooh. If there's not a wiki page on it, I get really mad. Because wiki will list the manufacturers for me. There's a psychology wiki fandom.
Fandom.
I guess this is literally it. Oh my god. I am floored.
Trade names. Methedrin.
Yeah. This can't be good. Meth is like uh oh my god. Oh, 14-hour shift. Good luck, my friend. Thank you so much for saying hi, hanging out, and following. You got this.
Sounds like you need some detox.
No. Give him hell. Yeah, dude. Wow, I'm so floored. I did not know that. Oh my god. Well, it's funny that we're talking about meth because we are about to talk about drugs. Because 5-HT2A is a psychedelic receptor. It is the main site that psychedelic drugs like LSD, shrooms, and DMT bind and mediate their action. So, I mean, and honestly, here's the thing: the best way to study a receptor is to do drug, and then, well, administer drug, and then you administer opposite drug to see if it blocks or increases the effects of the other drug. That's how we do it. So, for example, we know that LSD is an agonist at 5-HT2A, and that's what mediates its psychedelic effects like visual hallucinations and altered perceptions. The underlying mechanisms are a little, you know, unknown. I mean, serotonin binds this receptor all. It's happening right now in my brain. Serotonin is binding my 5-HT2A receptor in the visual cortex and in the PFC, but I'm not hallucinating right now. I'm pretty sure. I'm not hallucinating right now. So it's those exact molecular underpinnings that are so hard to ascertain. But we do know that when we apply an antagonist to 5-HT2A, those psychedelic effects go away. So even though we know that LSD can act at other serotonin receptors and even other non serotonin receptors, it's this antagonist specifically to 5-HT2A that abolishes the hallucinations. Pisses me off. I mean, fascinates me a lot. It it's a fun little tidbit, but it's kind of weird because the fact that we mainly understand this just because of the blockade of the receptor, like it's still actually not understood why serotonin doesn't cause hallucinations, but these other agonists do. But there is this is like the one thing I found, this observation that was made about LSD versus serotonin versus a Parkinson's treatment called Liceride. Um, all of these guys are agonists at the 5-HT2A receptor, every single one of them. But only one of them is hallucinogenic. Liceride and serotonin uh, only stimulate GQ, the GQ pathway. That's the one that's involved with calcium release. They only stimulate that pathway when they bind 5-HT2A. LSD, despite binding that literal exact same receptor in the same places, stimulates both GQ and GI, which I would imagine leads to a very stimulated cortex. Why though? Why? I don't know what it is about the structures of serotonin versus LSD that cause the receptor to activate different signaling pathways. It's just what the fuck. Um, MDMA was another ecstasy, Molly, is another serotonergic drug. It is not technically psychedelic, although I would describe it as a sort of combination between an amphetamine and a psychedelic. It doesn't it doesn't tend to cause hallucinations, but it does cause your neurons to totally dump their stored monoamines. I'm talking serotonin, I'm talking dopamine, I'm talking norepinephrine. Um, and if you've ever done Molly, then you may have experienced the next day you are like the most depressed you've ever been in your entire life. And that is because the it's kind of like the same logic as the therapeutic lag in SSRIs that I was describing earlier, but essentially you have this massive spike in serotonin, and that's going to upregulate these autoreceptors. They're like negative feedback, and they're going to overcompensate because there's a fuck ton of serotonin. That the next day you have lots of these autoreceptors and no serotonin, so you are very depressed.
I took, I did it, and but I took a particular what do you call it? Uh, su-
It
supplement.
was a supplement. Do you remember what it was?
Oh fuck.
It was like N something something. It was when I heard it, I thought that it was maybe something that your body could turn into serotonin. So taking it before doing ecstasy means that your body will have like this backlog of like probably tryptophan or something to make serotonin and prevent the downfall. Uh,
Acetyl L-carnitine.
oh yeah, okay. So that's part of the I believe. Why don't you look it up though? I believe that's part of the pathway that could potentially uh make tryptophan.
You can get it at Whole Foods. But that kept me from being extremely depressed. But it like I'll say that like my motivation was weird.
Oh
Like my
yeah.
sense of motivation, my sense of focus was weird. It was like normally focus is kind of like a fairly linear curve. Let's say curve.
Yeah.
It felt like I was driving on a road with bumps in it. Like I'm doing the thing I'm doing, but I'm just like like there's something that's just
Yeah.
like what the fuck. And
Well,
then like
focus is more of a
yeah.
dopamine thing,
Mm-hmm.
and I think the supplement that you took was more to replenish serotonin specifically, and maybe not all of your other monoamines. There is gonna be a test saga. No, I'm just kidding. Although I have been thinking at the end of one of these, I want to do like a test for a prize. That's been going around in my brain.
It helps the body turn fat into energy. Part of that.
Oh, okay. So,
Made in the brain.
well, that's still a big thing because drugs deplete your brain of its energy,
Mm-hmm.
and like ecstasy is just specifically associated with extreme depression, and I think maybe other psychedelics might have something like that. Like everything has like a come down, like a sort of hangover, but like people have described ecstasy hangovers as like you were the happiest you've ever been and you'll just never be that happy ever again. So like that is a very like serotonin kind of thing. So I think that not having energy though in your brain to have cognitive functions, depression is the lack of cognitive function in a
Yeah.
sense.
Having felt what it's like to like dance with it, it makes very much sense how I felt afterward. Like
Oh yeah, you
because I was
probably
so like, oh, I'm gonna dance. I don't care if I can or not. I'm going to
yeah.
do it, and I'm gonna feel good about it. And
Drugs are bad, kids.
yeah, well,
True.
drugs can be bad.
Drugs can be bad if you are not
Yeah,
if you don't prepare.
I don't go dancing like this
Yeah.
every night.
But this is similar to this like when you go to the dentist and you get a cavity filled, you have to be careful when you're chewing because you're numb. So it's like when you're on drugs, you feel like you can do anything. You're like, I can literally dance all night, and then you do, and then the next day it's like, oh my god.
It was a mix of like my my arms and legs didn't feel like they weighed anything, so like dancing felt easier. Like I wasn't straining myself to make movements,
Yeah.
but also like psychologically, like no sense of embarrassment, no sense of like
Yeah. Oh
self
yeah.
consciousness. Like
Yeah.
I'm going, you let loose and you're able to do it without thinking about it. Like it's good. It was
Yeah.
good.
That is good.
Um,
Like vegetables. Oh my god.
yeah, but yeah,
Drugs and
like
vegetables are both evil.
look up supplements if you're going to do that. What you should do because it's
Yeah.
going to make your life a lot easier afterward. Because drugs also can hurt you.
Oh yeah.
Yeah.
The rest of this, I think I have 2.5 more slides. This is all off the cuff. Uh, this is off the cuff because I was researching and a lot of things were inconclusive. So the rest of this is my personal thoughts and education. These are not definitive things. I'm not a doctor anyway, so you never should, you should always take things that I say with a grain of salt, but these more so are less solid information and more um, I am trying to process things. For example, hallucinations. Why? Why hallucinations? It's we don't really, it's weird because you would think that like, oh, but like dreaming is kind of like hallucinating. It's not actually. And serotonin is famously, you have no serotonin in your system while you are in REM sleep, which is typically the stage of sleep where people are dreaming pretty vividly. But you got no serotonin, so dreams are very distinct from hallucinations. Um, and I was reading that 5-HT1A and 2A are both expressed in specific parts of the visual cortex. And it was really hard to understand, but what I got was that neurons that are consistently firing at a low rate, like, they're neurons that are associated with things that should be outside of your attention, if that makes sense. So the rate of fire is proportional to how in your attentional sphere it is. HT2A stimulation tends to increase the firing of those neurons that are associated with more like outline and contour and like not non-attentional, but sort of on the cusp of your attention. So an example is this. I forget what these are called. I think it's like a Kaneza triangle. But if you stare at it, the white triangle that's upside down in the middle is an illusion. Um, and as you look at it, it gets more and more and more pronounced. And I think the idea of this study that I read was that stimulation of those 5-HT2A receptors is what is helping you focus on the contours of what is the illusion, essentially. Um, and how that relates a little bit to visual hallucinations is the logic of having something that's not normally firing and then it's increased. It's I believe it's called like a bottom up effect. So normally with sight, like if I'm like looking at something, that's a top down. I have the outside stimulus is being brought to my attention and it's being integrated and it's going down into these like, memory structures. Maybe that's top down. But with visual hallucinations, there seems to be this idea that neurons can spontaneously, like just like in the with the 5-HT2A activation that brings the contours. What's happened is we've gone from neurons that are kind of meh, like, and it's normal. The meh is normal to like being more upregulated by the 5-HT2A activation. So with psychedelics, there might just be this activation that's happening in the visual cortex that is bringing other things to your attention. And they are things that are not there. Like in this hallucination, I can see a line. We can see the line of the upside down white triangle. If you just sit there and you kind of stare at it, the illusion makes it look like there is a thin line making the triangle, but there is not. It's an illusion. It's a hallucination. So I think that and they did this study. Most of the studies, by the way, that I read about this, little bits are on humans. It's mostly rats. This one, it was monkey. It was macaques that they looked at their visual cortices and their serotonergic expression, and they activated those receptors. I just thought that was really cool. Okay, just for a little bit, I gotta talk about some things that kind of suck, mainly suicide. So that's going to be the last part, and then we'll be good. But just fair warning, I'm bringing this subject up because those 5-HT2A receptors are really like noticeably increased in a lot of different mental disorders. Like it is, I think, increased in the prefrontal cortex of schizophrenic patients, which I think makes sense because if you think about what psychedelics do, how they kind of can make you feel like this oneness around you, or they there's a word for this and I forget. They kind of make you have associations with things that are meaningless. Those are um, those are pretty common symptoms of schizophrenia as well. And it's thought that 5-HT2A in the prefrontal cortex um, does that. Um, and that same study that I got the those models of anxiety and depression from, that same study was concluding that, oh, because serotonin is involved in aggression as well. That's one of the more specific behaviors and feelings that serotonin is associated with. So having no or very low serotonin, obviously we get depression from that, but we also get aggression from low serotonin. And I wish it said specifically like the areas that had low serotonin because I feel like that's important. But having no or low serotonin and therefore having this depression with an aggression phenotype is demonstrates the highest risk for suicidal behavior because suicidal acts, suicidal attempts, and probably suicide itself. I would consider all of those to be aggressive acts. I think we tend to view aggression as like anger at like an outside source or like violence at other people, but suicide is violence at yourself. Um, and so it makes perfect sense to me that very high risk individuals on just the serotonin level, we're not talking about receptors yet, but just at the serotonin level, having low amounts would be a very high risk for suicidal behavior. Um, and then again, we saw this in the models, but low serotonin can lead to anxiety and depression. Normal serotonin has no anxiety, aggression, or depression, and then high serotonin is where you get the anxiety models because you have too much activity and communication going on in your cortex. Um, this picture right here is demonstrating um, areas of the brain. Ooh, I want to look at it specifically. It's not areas of the brain that have low serotonin. Uh, it's for areas of the brain that are most commonly found to be involved in functional and MRI brain morphometry research. Okay, I was hoping it would have receptors, but that's okay. These are areas that are usually studied. Probably receptor density, probably specifically 5-HT2A receptor density is studied in these areas. And all of these, I believe, all of these are most of them are in the frontal cortex. And then we have, of course, this is the parietal cortex. And then I would consider this and this to be more in the temporal cortex, but these are all areas of like those very high brain functioning, and emotional regulation, and so aggression regulation, and depressive and anxiety symptom regulation that are all sort of potentially dysfunctional in individuals with like major depressive disorder or maybe even people who are victims of suicide. Um, and then 5-HT1A, that's another, it's mostly the 2A. People really, really like to focus on that one. But 5-HT1A, it lowers activity in the cortex, which, as we know, we don't necessarily want that, which means that there is this reduction of restraint and an increase in the risk for suicide. Because remember, we all remember 5-HT1A is an inhibitory receptor. So if you have an increased expression in certain areas of the brain, then you are inhibiting communication to those areas. And then this says that cognitive testing has been used to find that suicide attempters have worse inhibitory control and executive functioning skills. And then they report prefrontal deficits in suicide behavior, impaired decision making, and social assessment. These are all like higher order social cognitive functions. And then this other one up here talks about the serotonin transporter, which is where SSRIs mediate most of their function by inhibiting the transporter to keep serotonin in the synapse. So the transporter on uh, axons that are on serotonergic neurons, there are. Wait, what? Oh, this is there's not as much in depressed. Oh, wow. Huh. I thought that said something else. But that's okay. It still highlights the importance of these two receptors when it comes to this very like unnatural and misunderstood um, process. Um, oh, I thought there was one more thing I wanted to say. I don't want to end on this necessarily. Thought I had one more thing to say, but I really found the studies on like these autopsies of people who had ended their lives. There was such mixed results that it was kind of hard to make my own opinion on what might be going on in the serotonergic system of these individuals. But I will say that those two receptors, they were both increased, which is interesting because they do opposite things. So I wish that the studies went more in depth on the areas of the brain that they found the increases in, because it wasn't always that cut and dry. I do remember that there was more HT2A in the hippocampus. Um, that was interesting, but ultimately, the most interesting thing for me was it was the visual thing. It was this because I had no idea about that, and I really had to kind of pull out of my ass like, what are these people studying these monkeys trying to say to me about the firing of these neurons and my perception of contours. Like that was kind of how they worded. They were like the perception of contours, the perception of contours, and I was like, what the fuck are they talking about? And it wasn't until like 3/4 of the way through the study that they posted that they talked about these optical illusions, and I was like, oh, the perception of the contours, like it. So,
Alex G. Alex Gray did the.
oh yeah,
Uh,
psychedelic guy.
yeah, yeah. You see them on my screen. Kind of what it looks like to do drugs.
Yeah,
To have visual, like and like your brain like. It, I don't know how to describe it, but it looks like that.
and they tend to be like they're usually like very simple shapes too,
Yeah.
and so it makes sense that like artists who do psychedelics and they are depicting their hallucinations. It's like these repeated patterns and like very clear boundaries between the different shapes. I think that's really really cool. But yeah, this fucking picture is really what made me understand what these studies were trying to tell me about visual hallucinations. Um, I'm gonna I mean, I won't post a link. It's I think it's on my like Twitch page, but the first video that I ever posted on YouTube for like science, it's an introduction to molecular biology. And my big example is these mother effing enterochromaffin cells. It's all about these guys.
Those Bart Simpson cells.
These Bart Simpson cells. Yeah, exactly. Because the proteins that make them up perfectly define their function. The mechanoreceptors, the enzymes that make serotonin, the protein that releases serotonin from the vesicle. Oh my God, it's like the perfect example of structure and function. And then having the neurons to demonstrate like, the propagation of the signal, and then the vomiting center and the chemoreceptor trigger zone were just perfect ways to demonstrate like receptor mediated actions. It's so cool. It's my favorite. Next time we'll have to see what I'm going to talk about. I'll have a vote up tomorrow probably. I know that second place, and I'm still going to put it on the vote, but second place was about the diabetes. Diabetes mellitus and diabetes insipidus.
Insipidus drink.
I am going to have a sip of this drink. Well, thanks everyone. This was great. This was less painful than I thought it was going to be cuz boy, were these things complicated. I didn't even talk about 5-HT4, 5A, 6 and 7. Fuck those guys. Fuck those fucking guys. If I had to choose a favorite, it would be 5-HT3 because it's different. It's not a stupid G protein. It's a ligand-gated cation channel. Thanks for hanging, y'all. I will hopefully see you all tomorrow for Morrowind. Tomorrowwind. Deuces.